mynews.studio.
A single gene edit saved a baby's life this spring. Could this be a turning point for medicine and food?
Today is June 11th, 2025. Let's start with the story from last month. A 5-month-old named KJ received a personalized CRISPR treatment for CPS1 deficiency. That's a fatal genetic disorder, and the therapy corrected a single-letter mutation in the baby's liver cells. What really stands out is the speed, only 6 months from diagnosis to therapy. Does this set a new standard for individualized medicine?
It does. What's especially striking is how quickly KJ recovered, and so far there haven't been serious side effects. But there's a larger point here too. This was a coordinated effort between scientists, clinicians, and regulators, all moving fast. If it's possible for one child, is it feasible for more, especially in places with fewer resources?
That's the tough part. High development and delivery costs, limited infrastructure, and not enough trained staff. These are big obstacles in low and middle-income countries. Plus, regulatory processes can slow things down. Are organizations stepping into?
They are. Groups like the World Health Organization and the NIH are backing scalable gene editing platforms, and they're encouraging flexible regulatory pathways that can work in different settings. Still, even with their involvement, it's clear. Building infrastructure, training people, and ongoing investment are all necessary to make these therapies accessible in underserved areas.
Let's take a broader look at CRISPR clinical trials worldwide. There are around 250 ongoing trials, not only for rare disorders, but also for things like high cholesterol and hepatitis B. Some results are pretty striking. For example, a single-dose CRISPR treatment lowered oxalate levels by almost 70% in a rare kidney disorder. Another reduced bad cholesterol by 70%. Could this mean people might not need lifelong medication anymore?
It's possible. Early trials for hepatitis B are also promising. Researchers are seeing substantial drops in viral markers. If these results hold up, we might see one-time treatments replacing chronic therapies, but precision and safety are crucial.
On the topic of precision, the CRISPR toolkit keeps growing. Beyond CRISPR-Cas9, there's base editing and prime editing now. These allow for even more exact changes to DNA. David Liu's team at Harvard and other groups like those at Yale using CRISPR-Cas12a are taking this further, editing several genes at once in mice. Is this likely to change things for complex diseases like cancer or autoimmune conditions?
It already is. By editing multiple genes in one experiment, researchers can build better disease models and test advanced therapies. This speeds up progress for diseases that involve more than one gene or pathway, not just single mutations.
Let's shift to agriculture. Scientists are using CRISPR to make crops more resilient to drought, heat, and salty soils. For instance, CRISPR-edited tomatoes and maize are showing better drought tolerance and higher yields under stress. Why is this important right now?
Because climate change is putting food security at risk everywhere, crops that can survive harsher conditions could help us avoid future food shortages. And it's not just about survival. CRISPR can also improve nutrition by turning on helpful genes in plants. The potential to reshape our food supply is significant.
All this raises ethical and regulatory questions. For now, most CRISPR therapies edit only somatic cells, so the changes aren't inherited. But the technology can also alter the human germline. How do cultural and societal values influence how people accept this technology?
They have a major influence. In some regions, strong concerns about changing human DNA, especially if those changes could be inherited, have led to bans or skepticism. That's why global communication and policies tailored to local values are key to building trust.
Another important point, what safeguards are in place to prevent misuse or unexpected effects? We're talking about off-target changes or gene-edited organisms with unknown impacts.
Researchers are developing more precise gene editing tools, and they're running thorough preclinical and clinical tests. There are also international guidelines being developed, focusing on transparency and oversight. But as gene editing technology moves forward, the safeguards will need to keep evolving too.
So where does that leave us? CRISPR is already changing medicine and agriculture. For some people, like KJ's family, or farmers facing climate stress, it's brought real hope. But there's still a lot to figure out around safety, fairness, and what it means to edit life itself. Can we manage the risks and make sure everyone benefits?
That's the central question. The next few years will show whether CRISPR's promise can reach everyone, and if its risks can be managed responsibly.
We've covered a lot today. Breakthrough therapies for infants, gene-edited crops for food security, and the ethical and regulatory frontiers. The CRISPR revolution is underway, and it's global.
Thanks for listening and staying curious about the direction science is taking us. This episode was brought to you by mynews.studio. Try it now for free.